we are here today to talk about the genetic code, nature's own way of inscription and encryption. Basically, every living system is made up of cells, and as a cell is a single biological unit of life, every single cell contains within its nucleus the master molecule called DNA. This DNA contains a blueprint for all the kinds of characteristics that the cell or an individual is going to exhibit. DNA works via the agency of RNA. Different types of RNA molecules are there: messenger RNA, transfer RNA, as well as the ribosomal RNA. These RNA molecules collectively are responsible for synthesis of different types of proteins, which are working extremely specifically in controlling individual characteristics that are exhibited by an individual. The manner in which DNA works through the agencies of RNAs, the RNA which carries the message from the DNA to the cytoplasm regarding the kinds of proteins to be synthesized is known as the messenger RNA. This messenger RNA contains a sequence of nucleotides which is regulated by the sequence of nucleotides on the DNA molecule. And the sequence of nucleotides on the messenger RNA determines the sequence of amino acids that a synthesized polypeptide chain will contain. Looking to this, a messenger RNA contains the message regarding what kind of protein has to be synthesized. Without going to much detail about how exactly does a messenger RNA work, we have to bear in mind the fact that every triplet of nucleotides on messenger RNA is responsible for placing one amino acid in a polypeptide chain to be synthesized. And these triplet of nucleotides has been called genetic code, which, let me repeat, is responsible for synthesis of a polypeptide chain, a particular amino acid. Where a amino acid will be placed, in what sequence it will be placed, is determined by the sequence of nucleotides on the messenger RNA. Every triplet of nucleotides is responsible for this, known as genetic code. With this primary information, we are going to now elaborate upon genetic code, and I request Dr. Bhupesh Yagnik to please take over.
Thank you very much, Piyush Bhai. As it was described by him, that every living being has a blueprint of genetic information encoded in the form of DNA. And every individual is so in terms of characteristic, because the genetic information contained within each and every cell defined so. Now, this information which reside over a sequence of DNA is in fact read in a very specific manner within a biological system. And this language, a secret language of nature, was deciphered by scientists by working years together in a painstaking efforts. And only after these efforts, the secret language of nature was revealed. Now, in today's lecture, I am going to tell you about a detailed account on how these discoveries were made and what kind of experimental tools were used by this scientist, which led to won the Nobel Prize in medicine and physiology. So, starting with a concept of central dogma, when John Gregor Mendel explained the laws of inheritance of a trait, the idea behind understanding these nature's language were routed. And especially after the work of Frank Griffith, who described that genetic material is in fact a DNA, the molecule received widespread attention by the scientists worldwide. And in an efforts to characterize the structure, Watson and Crick has described the double helical structure. And it was the work of Beadle and Tatum in 1958 who described that the information within a DNA is actually expressing itself by means of protein. And for every segment of a nucleic acid, there are specific proteins which are made in accordance with the sequences, and these proteins are responsible for executing themselves and imparting a specific trait or characteristics. Afterward, it was Charles Janowski in 1960 who described that a definite kind of collinearity is existing between the sequence of DNA and that of a protein. Because after 1950, especially the sequence of protein named insulin was characterized by Sanger, a lot of information were available all about the sequences of nucleic acid and protein. And these sequences were correlated by Janowski, and he explained that a definite correlationship is existing in terms of sequences of nucleic acid and proteins. And that is how the efforts to understand and decipher the genetic information of a DNA began. Now let us have a look at how this nucleic acid information is expressed in the form of protein. This happens when messenger RNA expresses itself into a protein during a translation process. Now what I am going to show you is a small animation which describes how proteins are made inside a cell. What you see in this animation is messenger RNA strand binding itself to two ribosomal subunits at an appropriate site, and from a point from where the translation is to begin, tRNA binds, and this tRNA carries a correct type of amino acids. When two such tRNA brings together two amino acids side by side, peptide bonds are formed between them, and the process goes on. Every time new tRNA brings in new amino acid, and that is how a length of polypeptide chain grows inside a cell till it acquires a three-dimensional functional unit as shown in the cluster, and such a protein is sent out to carry out its function. Now, this process can actually happen at a very fast pace. What we are seeing in this diagram is a nuclear membrane having a pore through which the messenger RNA is about to come and reach into the cytoplasm. And within this cytoplasmic region, the messenger RNA is translated in a rapid manner. Green colored triangular shaped molecules are in fact tRNAs, which are constantly bringing in new amino acids, correct type of amino acids, in accordance with the nucleotide sequences over the messenger RNA. And these amino acids are joined together until a functional polypeptide is synthesized and released into the cytoplasm. The process continues till every information within the messenger RNA strand is encoded into right kind of protein. Now, how this language of messenger RNA is interpreted was a very big challenge to the scientists, and I am going to tell you about the work carried out by these three scientists, namely Robert Hawley, and Hargobind Khurana, and the Marshall Nirenberg. These three scientists were the winners of Nobel Prize in Medicine and Physiology in 1968 in recognition of their work on deciphering the genetic code. Now, the experimental approach used by this scientist was very much challenging. What they have done is they resorted to several kind of experimental tools and approaches to fully understand the language of nature. The problem like riddle of solving the genetic code seems to be very trivial in the present day scientific know-how and technological expertise that we have right now. Today, deciphering all 64 number of genetic code may be a small exercise taking hardly more than 24 hours by use of the advanced instrumentation technique like gene sequencer, protein sequencers, and all kind of automated instruments. But way back before 1950 and after 1960 in particular, the scientific know-how and technology was not that much advanced. The major drawback was availability of the test RNA sequences, and this was availed by the work of Nirenberg and Ochoa when they described for the first time enzyme called polynucleotide phosphorylase. Now, this enzyme was described from E. coli, which does not require any primer and can polymerize nucleotides to a small length of RNA. And this kind of synthetic RNA molecules in fact served as an important tool in the entire experiment by various approaches. Second important tool used by these group of scientists were development of in vitro protein synthesizing system. And within this system, they run several kind of experimental design, especially homopolymer RNA translation. Then RNA repeating co-polymers were synthesized and were translated and studied in the same set. And randomly co-polymerized nucleotide sequences were also tested. And most importantly of all, most effective and most powerful approach was ribosomal binding assay developed at a subsequent stage, which probably put an end to the problem of deciphering the genetic code. Now we will understand each and every experimental approach one by one. The cell-free translation system, which was developed by Nirenberg and his colleagues, is in fact a very simple material. E. coli cell mass was actively grown and it was ground, and all the cytoplasmic contents were separated from the cell debris, like membrane component, wall components, etc. Now what remains in the supernatant is in fact an extract which contains all kind of ribosomal subunits, then all kind of tRNA species properly charged with right kind of amino acids, all enzymes and factors which are responsible for synthesis of protein. These were there in the cytoplasmic residue. The characteristic of cell-free translation system is that it is inefficient. That is not as rapid and as efficient as the in vivo cell translation system. But it has some advantages, although it is short-lived, but does not require specific initiation sequences to begin the translation process. Such a system can be made into initiation of a translation process simply by manipulating high level of magnesium concentration. Within this system, one can readily detect the newly synthesized polypeptide molecules. After that, the second approach was use of homopolymer testing. Within these cell-free protein synthesizing system, what they have done is they artificially synthesized polyuracil chain. A artificially synthesized messenger RNA was made by them, which contains only one nucleotide, and that is uracil. So this poly-U messenger RNA species was fed as an artificial substrate to the protein synthesizing system to be run in vitro condition. What they have observed is only one kind of amino acid, that was phenylalanine, which was polymerized. So the resultant product was protein which contains only one amino acid, and that was phenylalanine. So the language was simple, that U type of sequence leads to amino acid phenylalanine. In the same way, they were able to describe and understand the meaning of poly A codes, poly C codes also, which were coding respectively for amino acid lysine and proline. But when they carried out similar experiment with poly G type of messenger RNA chains, they observed that the artificial substrate mRNA was undergoing heavy enfoldings, so could not be translated successfully. So with these experiments, the precise meaning of UUU was not understood. For that, a second approach came to their help, and that was using repeating copolymers. what they have done in these experiments is they mix up two kind of nucleotides together, namely uracil and cytosine, and they allow them to undergo alternating, get polymerized in alternating manner. That is UC type of sequence was made. And when these chain was translated under in vitro condition, they got a polypeptide which was having alternating arrangement of two amino acids, serine and leucine, serine, leucine, serine, leucine, likewise. Although it could help us in understanding that there are two types of codons possible, UCU or CUC, and both of these codon might be coding for either serine or leucine, but the precise information was not available. The third approach was using random copolymers. They selected two kind of nucleotide monomers, namely A and C, mixed them in different ratio, like in 50-50% ratio, in 10% or 90% ratio, and they calculated the net frequency of occurrence of specific codon sequences, like as shown in this diagram, probability for CCC, CCA, CAC, likewise. This can be calculated statistically. And when these type of polymer, namely randomly co-polymerized polymer, messenger RNA, when these was allowed to undergo translation in the in vitro condition, they obtained several amino acids, and these amino acids were proline, asparagine, glycine, histidine, threonine, and lysine. Now, two codons, especially CCC and AAA, were deciphered in the beginning. So, rest of the codons might be coding for one of these four amino acids, but the exact meaning of this was difficult to derive out of such an experiment. So, the entire challenge of understanding the genetic code came to the end when ribosomal binding assay was developed by two scientists in the laboratories of Nirenberg himself. Now, this scientist, along with the help of Philip Leader, developed a process in 1961 called ribosomal binding assay. With these assay, the entire challenge of understanding the genetic code was conclusively ended. This experimental approach is based upon use of specific kind of filters called nitrocellulose filter. These filters are having a very fine porosity and has a tendency to retain large molecular weight substances, allowing small molecular weight materials to pass through. So, in other words, larger aggregates like ribosomes are retained over the membrane because they can't pass through the membrane, while small molecules like tRNA, amino acid, enzymes, and small proteins, messenger RNA, such small molecular materials can easily pass through the membrane. Now, what they have done is, as shown in this animation, he allowed messenger RNA and ribosomal aggregates to pass through the cytoplasmic membrane. And you can see that large aggregates like ribosome, messenger RNA complex were retained by the filter. Afterward, the messenger RNA attached to the ribosome were a small triplet of a known sequence, which were chemically synthesized in the laboratory, especially after the Hargobind Khurana joined the team of Nirenberg. The tRNA species which were charged with different kind of amino acid, when these was allowed to pass through the membrane, it will not be able to establish itself over the messenger RNA because the sequence specificity does not exist. But when tRNA, which is having a compatible anticodon arm, is allowed to pass through this membrane, it will establish itself to messenger RNA because sequence compatibility exists. And when this experiment is repeated 20 times, every time one of the amino acid was radioactively labeled, he was able to understand that if radioactivity is retained in one particular amino acid set, that amino acid is probably encoded by the sequence under the experiment. So although it was a cumbersome and tedious task, for every nucleotide tested, he has to carry out and run number of sets every time, starting with E. coli cells and preparing a fresh extract of them and a fresh cell-free system until the proteins are checked up. But it eventually put an end to a challenging task of understanding the codons. And with these experiments and years of efforts by a team of scientists, entire dictionary of genetic codon became known to us. And you can see over in this chart that all amino acids are coded by one or more number of genetic codes. There are specific sequences as shown in the green called AUG, which codes for initiation of a process, and those sequences which are shown in red, UAA, UAG, and UGA, are responsible for putting an end to the translation process, are known as termination sequences. The rest of the codons are known as sense codons because they are coding for one or the other type of amino acid in particular. Now, how these genetic codes are read, what are the rules of this language, are there any punctuation marks or not, are these sequences overlapping with each other? A lot of questions remain unanswered, and the efforts were also going on side by side along with the understanding of the codon. And now I'll hand over to Piyush Bhai for explaining the general characteristic of this codon and the rules and grammars of the language spoken by nature. Piyush Bhai, please.
So once the clarity has been established, hopefully, among the students regarding the basic technology that brought to us knowledge the structure of the code and the constitution of genetic code, now let us capitalize upon certain characteristics which, let me make very clear in the beginning, that are universal. The characteristics of the genetic code are universal in the meaning that from a small bacterial cell to a plant or an insect or an animal to us, the human being, the genetic code remains almost the same. We'll be talking about that as well, but the characteristics by and large remain the same in all types of biological systems, be it a prokaryotic system like a bacterial cell, or be it a dinosaur, which though have become extinct, in their time would have followed the same rules and regulations regarding the genetic code's existence and its activities. If we may have the slide, please. The first thing that strikes us is that genetic code is made up of 3 letters, 3 nucleotides. What is the logic behind this? As we know, the nucleic acids, the messenger RNA which carries the message from DNA to the cytoplasm regarding the kind of protein to be synthesized, is made up of nucleotides, and by and large in a given nucleic acid we get 4 different varieties of the nucleotides. In a nucleotide structure, the sugar and the phosphate remain the same. The only variation possible is among the nitrogenous bases, and by and large, as we already know, 4 different types of nitrogenous bases, the nucleotides containing either adenine, which is called an adenosine nucleotide, and similarly guanosine or thymidine or cytidine kind of nucleotides are available. So we have at our disposal 4 nucleotides, out of which we have to prepare the codes. Codes will have to specify to the cytoplasmic protein making machinery what kind of amino acid has to be placed where. Now there are 20 different types of amino acids which are found in naturally occurring protein molecules. So 4 nucleotides have to give information for 20 different amino acids. Naturally, if we assume that the genetic code is made up of only a single nucleotide, then a single nucleotide code will be able to give information for only a particular amino acid, a single amino acid, and therefore only 4 amino acids can be specified. If we buy the fact that the code is made up of only a single letter, only 4 amino acids can be specified. If we assume that, okay, let the code be consisting of 2 nucleotides, then what will happen? 4 nucleotides, and every single code made up of 2 nucleotides at a given time, then the total combinations possible is 16. So still we fall short by 4 amino acids. 16 different genetic codes made up of 2 nucleotides can give information for 16 different amino acids, but what about the remaining 4? In that case, the next probability is that the code should be made up of 3 nucleotides, adjacent neighboring nucleotides. Given this, total number of codes available goes to 64. Now the problem is there are 64 different code combinations and we have 20 amino acids to specify. Now, as we know, nature is a great economist. There is no extravagancy among nature. And if we go by this equation, for 20 amino acids, we have 64 different codes. So an extravaganza of 44 codes exists. And this is not acceptable. To the common sense of a scientist, this kind of extravagance is exorbitant and cannot be tolerated. But then the fact remains, as we'll go on talking about the characteristics of the genetic code, that this justifies the total number of 64 possible codes for 20 amino acids is quite justifiable. As a matter of fact, it is quite beneficial to a biological system. We'll be talking about that later. At the moment, let us have the next slide, which tells us the known overlapping property of the genetic code. As I told you only a little while ago, the genetic code is made up of 3 nucleotides, and these 3 nucleotides have to be adjacent to each other. The neighboring nucleotides on a given messenger RNA sequence, the 3 neighboring nucleotides make up a single code. Now, if we understand, let us assume for a little while that the code is overlapping. What do we mean by that? If you look at the figure, you can see a sequence of GCC, CAC, and if we buy the fact that the code is made up of 3 letters, it gives us 2 genetic codes, GCC and CAC, where GCC, as you can see in the diagram, codes for alanine and CAC codes for histidine. Okay, if we accept that the genetic code is overlapping, then what will happen? In this sequence of 6 nucleotides, we can have a possibility of 4 genetic codes, the first code being GCC, then the next can be CCC, then the next can be CCA, and the fourth one can be CAC. Thus, as I told you a little while ago, nature is a great economist. It can conserve here by making a compact code structure where the 6 nucleotides in this figure can give 4 different codes. This is what an overlapping code is consisting of. But the fact remains that there is no overlapping. There has to be a demarcation, there has to be a differentiation of 3 nucleotides each, and there has to be no overlapping. This was proved very easily by an experiment where a change in any of the single nucleotide in a given sequence would affect only a single genetic code. If there was overlapping, for example, look at the third nucleotide in this given sequence, GCC. If this C is replaced by some other nucleotide, for example G, then the first code changes. GCC now turns into GCG code, and this gives information for tyrosine. So alanine will be replaced by tyrosine. Now the next code also should change. If the genetic code is overlapping, then the C being replaced by G will change the third code as GCA, and this will change another amino acid as well. So a single mutation, a single change in the nucleotide sequence will at a given time make two changes in the polypeptide. Two amino acids would be replaced by undesirable amino acids. This doesn't happen, and therefore it has been confirmed, it has been established that code is never overlapping. There are certain exceptions in viruses where some kind of overlapping is done, but as we understand, they are rare exceptions. By general rule, in almost all biological systems, the code remains non-overlapping. The next slide tells us about the start and the stop signals. Messenger RNA is actually made up of a reading frame. It is called an ORF, the open reading frame, where the stop signal, first of all, the start signal is there, where the ribosomal assembly understands that this is the point from which the initiation of translation has to be made, and then there is a stop signal as well, where the whole process of translation, the synthesis of a polypeptide comes to an end. The initiation codon in most biological systems is AUG, and it always places as the first amino acid of a polypeptide methionine. The protein's translation machinery has been so established that in any given polypeptide chain, the first amino acid to be placed is methionine. In prokaryotes, this methionine is formulated. In eukaryotes, the formulation may or may not take place. We will not go into details of that. But in most biological systems, as the preliminary, as a primary genetic code, we have AUG, and that always invites methionine as the first amino acid of the polypeptide chain. Similarly, when the chain has to terminate, the whole process of translation has to come to an end. For this, there are three different genetic codes. These codes, UAA, which is called Osher, UAG, which is called Umber, and UGA, which is called Opal. It is also called Umber. They have a very specific function of bringing termination of the polypeptide chain synthesis, translation process, and thus there is no tRNA which is specific for them. They never invite the amino acids for translation machinery. Rather, they tell the translation machinery that this is the point from which the process has to terminate, and the ribosomal assembly has to be separated from the messenger RNA. So let us remember that basically there are 64 different combinations of genetic codes, out of which 3 do not code for any amino acids. So for amino acids, the SANS codon number is now reduced by 3, 61. We'll be talking about how this 61 number is justified for 20 amino acids later on. But at the moment, it is very important to realize that out of 64, 3 have a very important function of bringing the polypeptide synthesis to termination. The next slide tells us about the degeneracy of the genetic code. Degeneracy or the redundancy is a feature of the genetic code where a single amino acid can be encoded by more than one genetic codes. And this tells us exactly why there are 61 codes for 20 amino acids. By a simple logic, the messenger RNA and the DNA molecule, which is responsible for synthesis of messenger RNA, are always in a dynamic state. That is, the DNA is always being threatened by a phenomenon called mutation, where the nucleotide sequence of DNA can undergo change. A mutation is always permanent, inheritable change. And once the mutation is established, it can immediately be reflected into the respective messenger RNA molecule. If the mutation is reflected into the messenger RNA molecule, there is always a probability that the sequence of nucleotides on the messenger RNA, which is acting as the code, will undergo a change, and an undesirable amino acid can be placed in a resulting polypeptide chain. Once a wrong or undesirable amino acid has been placed in a growing polypeptide, the polypeptide may not function in the desirable manner, because the folding and thus acquiring the three-dimensional shape by a polypeptide chain depends largely upon the number of amino acids and sequence of amino acids within it. So a single amino acid shift in a polypeptide can affect the three-dimensional configuration of the resultant polypeptide in a very bad manner, and this polypeptide either will be totally unable to carry out the function for which it is meant, or if at all it carries out the function may not progress in the desirable speed. Therefore, it is always desirable, it is always in the advantage of a biological system that the fidelity of translation, that is the placing of a amino acid at a particular position, should be very faithfully maintained. Now let us assume that one particular position has changed, a particular nucleotide has been modified by a mutation at the level of DNA, and because of this there is a chance that the desired amino acid may not be placed in the proper place. If the changed genetic code carries information for the same amino acid, that is an advantage. But is this possible? It is indeed possible. There are many amino acids which have more than one codes. As a matter of fact, there are three: leucine, serine and arginine. These three are the amino acids which have as many as 6 genetic codes each. You can see in this chart that arginine and leucine have 6 different codes. So any of these codes in a messenger RNA molecule will give information for the same amino acid: serine, arginine and leucine. Similarly, threonine, proline and many others have 4 codes each. Isoleucine has 3 codes, and there are many out of 20 amino acids which are encoded by 2 different codes. Methionine and tryptophan are the only amino acids which are being encoded by only a single amino acid. The remaining amino acids have the luxury of having more than one codes, and this makes up for the probability of the mutations at the level of DNA, which may be ultimately transferred into the messenger RNA, reflected into the messenger RNA. So the redundancy or the degeneracy of the genetic code, that is a single amino acid being encoded by more than one codes, is a very good advantage, and this I hope will now justify very properly the fact that for 20 amino acids there are 61 different codes. This makes up for the probability of the mutations. This is a very good counter mechanism against the possibility and probability of the mutations occurring randomly at different positions in the DNA and then being reflected into the messenger RNA. So this is a very important property of the genetic code. The next property of the genetic code is, as we talked in the beginning, the universality. From a simple bacterial cell to a large elephant, all types of biological systems, microbial or otherwise, have the same genetic codes. If AUG codes for methionine in a bacterial cell, it codes for methionine in human being, us as well. The only exceptions, for example, are the mitochondrial protein synthesis. We know that the mitochondria have their own protein synthesis, they have their own machinery. The entire mitochondrial protein synthesizing machinery is independent of the cytoplasmic protein synthesizing machinery, and the start signal in mitochondria, that is for AUG, which is coding for methionine in other biological systems, it is GUG in mitochondrial system. Some very early forms of bacteria, which are called the archaebacteria, may also have some kinds of exception in genetic codes, but otherwise for almost all biological systems, as you can read in this particular slide, that from guinea pig to mice to E. coli, anywhere you check and you find the same start signals, same stop signals, and the exception being the mitochondrial DNA. The plants have chloroplast as well, and in certain chloroplast, in the chloroplast of certain plants, once again, the start signal is GUG in place of AUG, but all plants don't have this kind of sliding away from the basic rule. Most plant chloroplasts have only AUG as the start codon. So this is how, these are the basic characteristics of the genetic code: the universality, then the redundancy, then the non-overlapping. In a similar manner, one more property of genetic code is that it is not punctuated. This is very important to understand, the non-punctual nature of the genetic code. When we look at messenger RNA sequence, every triplet in a continuous manner will be acting as genetic code. What we intend to tell you is that there are no in-between sequences where no meaning of that particular portion is there. In a continuous manner, the entire messenger RNA nucleotide sequence is translated into the polypeptide. Another very interesting property of the genetic code is Wobble Nature. This is a Latin term. Wobble is actually the actual meaning of wobble is a wave, the manner in which a wave progresses. When a combination between two different systems is not very stable, that combination is not allowed by the nature. And we know that during protein synthesis, the messenger RNA has to produce the triplet of nucleotides on the messenger RNA. The genetic code has to make a stable contact with the anticodon part of the transfer RNA, which carries a specific amino acid. Now, triplet on the messenger RNA is consisting of 3 nucleotides, 1, 2, and 3. Transfer RNA, on the other hand, will have an anti-parallel arrangement, that is, against the first nucleotide of the messenger RNA, the third of the transfer RNA will be placed, 2 and 2 and 3 and 1. This is how the combination is. Now, the third letter of the messenger RNA sometimes may have changed because of the mutation or for any other reason, due to any other reason. If this third letter of the messenger RNA code is not proper, then the combination between the code and the anticode of the transfer RNA is still established. First 2 nucleotides of the genetic code and second and third nucleotide of the transfer RNA can make hydrogen bonds against each other as shown in this figure, and any deviation in the third letter, you can see it should have been C, but it has been replaced by U. Transfer RNA as is first nucleotide G, which would have made hydrogen bonds against cytosine of the messenger RNA, the third letter of the messenger RNA. But this cytosine has been replaced by U. So naturally the hydrogen bonds are not going to be possible between U and G of transfer RNA, but still you can watch the U and A of messenger RNA and tRNA, and then C and G of messenger RNA and transfer RNA respectively have been able to establish hydrogen bonds, and as a result of this, the proper hydrogen bonds between first 2 nucleotides of messenger RNA and second and third of transfer RNA, a proper binding is established, and the intolerance of the third letter of the messenger RNA and first of transfer RNA is nullified. This is not a proper binding. I would exemplify by imagining yourselves riding, or rather standing in a crowded bus where you are not allowed to put both your feet planted on the platform of the bus. You will hold the upper handle of the bus and you will somehow stand on a single foot, making a wavy movement as the bus takes you ahead against the passengers who are also sharing the same misery as you are. This position is wobble, and genetic code and anticode of the transfer RNA are tolerating some modification in the third letter of genetic code in the same wobble manner. So this is also a very important characteristic of genetic code, the wobble nature. With this, we have come to the conclusion regarding the nature of genetic code and the characteristics of genetic code. You can have a glance at the references, and in the meanwhile, if we have any telephone call, welcome. If there are no telephone calls from both of us, Dr. Bhupesh Yagnik, who is largely responsible for making of the presentation, he is the one who has prepared all the slides, the what you call the animations as well have been prepared by him. And we once again thank you all, the Commissionerate of Higher Education, the BISAC building, the staff of BISAC, and all you who have watched us. Thank you very much.